Okay, so our next topic to discuss is blood. So blood is a connective tissue and it's important to transport vital substances through our body. We are going to learn about the different components of blood. It is important to regulate homeostasis. It's also important to distribute heat. And we're going to talk about the specific parts of the different blood cells. So there's red blood cells, white blood cells, and platelets. And we're going to see that the amount of blood varies with body size, changes in fluid concentrations, electrolyte concentrations. Typically, blood is about 8% of an adult's body weight, and adult blood volume is about 5 liters. So here is a sample of blood being processed, and in this example you can see the blood is being centrifuged or spun down, and once that happens you see it is in these 3 distinct layers. So there's the plasma, there's this part in the middle called the buffy coat, this is the white blood cells and platelets, and then down here at the bottom are the red blood cells. So we're going to look at the different components of blood, this capillary tube, and then it shows the separation of the different components of blood. So on this slide, it shows that blood is about 55% plasma and about 45% what they're calling formed elements. So these formed elements are red blood cells, white blood cells, and platelets. It shows that these formed elements, it's about 95% red blood cells, about a little less than 5% platelets, and about 0.1% white blood cells. Now, in this 0.1% of the white blood cells, there are 5 different types of cells that then can go on to be different cells of the immune system. We're going to talk about these a lot more later. In the plasma, while this doesn't contain any of the red blood cells, white blood cells, or platelets, it still has a lot of necessary vital things for survival. So it contains electrolytes, different ions. It's about 92% water, about 7% proteins. There's albumins, globulins, fibrinogens. We'll talk about what each of these do. It contains waste, it contains different nutrients, vitamins, hormones. Remember, in the endocrine system, we said hormones go through the bloodstream, so this would be where those hormones are, in the plasma part of our blood. And then also gases. So there's nitrogen, oxygen, and carbon dioxide in our blood plasma. We will really mostly focus on oxygen and carbon dioxide. Oxygen is the molecule that we need for what's called cellular respiration. That's the process by which we make energy, we make ATP. And then the waste from that process is carbon dioxide. So nitrogen is in the air that we breathe. It's actually the most prevalent gas in air that we breathe, but it doesn't really have a physiological role. So here's another breakdown of the blood cells. So we're going to talk about each of these in more detail, and this is just kind of a good schematic to show how each of these are going to give rise or where they come from. So again, really red blood cells, white blood cells, and then we'll talk about platelets a little bit later. So the origin of the blood cells, they originate in what's called red marrow, and these are from hemocytoblasts or hematopoietic stem cells, meaning these are the precursor to all of the different classes of blood cells that we just looked at on that previous slide. So some stem cells, these hematopoietic stem cells, to be a stem cell just means that there's the potential to differentiate into another cell. So some of these stem cells will differentiate into more stem cells. Some of them will differentiate into lymphoid or myeloid stem cells, but we're going to look at each of them individually. So red blood cells, these are erythrocytes, and a red blood cell is described as a biconcave disc. So it kind of looks like a bagel without a hole all the way through the middle. And the reason for this shape is to increase the surface area. So surface area is a really important characteristic of a lot of the cellular structures that we'll look at throughout anatomy and physiology, because most of the time the chemical reactions that we're looking at can only happen on the surface of that structure. So we want to have the most surface area on this red blood cell because it's on the surface of the red blood cell where this gas exchange is going to happen. We'll talk about this a little bit more later, but the role of a red blood cell is to transport oxygen. So oxygen is not soluble in our bloodstream, so this is back to chemistry, but remember that our cells, our body is mostly made up of water, and water is polar and oxygen is nonpolar. So polar things and nonpolar things do not mix. So we can't just breathe oxygen into our lungs and then have that oxygen freely diffuse through our bloodstream to all of our organs and tissues. So for that reason, we need a special molecule to carry the oxygen from our lungs to all of our tissue. And the molecule that does that is the protein called hemoglobin. And a red blood cell is basically just a cell that is full of hemoglobin. Red blood cells actually eject their nuclei and mitochondria once they're fully developed to make more room for more hemoglobin. So what a red blood cell does is it's just full of hemoglobin, and it picks up oxygen in the lungs, and then it brings the oxygen to all the different parts of our body. And again, the reason that it's shaped this biconcave shape is because that maximizes the surface area, and the places where it can pick up the oxygen is on the surface of this molecule. So we have two different forms of hemoglobin. We have what's called oxyhemoglobin, which means this hemoglobin has picked up an oxygen. And then we have what's called deoxyhemoglobin, meaning there's no oxygen attached to this hemoglobin. So we're going to talk about all of this a little bit more later. But red blood cells, it's the most prevalent, most highly concentrated cell in our blood. And again, the purpose of the red blood cell is just to transport oxygen. So here's a little bit of details about red blood cells. As we go through all of the different types of cells, we're going to see the percentages and numbers of these cells, and then there will be a table at the end that's going to compare all of them, so don't worry about this for now. But a red blood cell count is the number of red blood cells in a cubic millimeter or a microliter of blood. So this depends on your age and your health and your sex, male or female. And what a red blood cell count reflects is the oxygen carrying capacity, because again, the job of the red blood cell is to circulate oxygen from the lungs to the tissues. So this is showing erythropoiesis, and this is showing new blood cell formation or generation. A red blood cell can only live for about 120 days, and this is the process by which we would make new red blood cells. So when there's low blood oxygen, meaning we're not transporting enough oxygen as we need, it stimulates this protein called erythropoietin, EPO, and this stimulates more red blood cell production, and then we make more red blood cells so we can carry more oxygen. This EPO is actually one of the performance enhancing drugs that is tested for in athletes. I think this is the one that Lance Armstrong was caught using, where it increases the athlete's ability to transport oxygen because they're increasing their body's natural red blood cell production. So this is one of the performance enhancing drugs that are tested against. If you have an increased EPO level, it's probably because you're taking a drug that is causing that to happen. Let's see. So for this erythropoiesis to make more red blood cells, we need vitamin B12 and we need iron. And we get this from the food that we eat, from dietary sources, and we'll talk about this more when we talk about the digestive system. Vitamin B12 and folic acid are necessary for DNA synthesis, so this is important for red blood cells, but also making all types of cells. Iron is important because iron is the metal that's at the middle of this heme ring that's at the middle of hemoglobin, which is the protein that's inside the red blood cell that's carrying the oxygen. So we need the iron because that's what the oxygen will actually bind to, and that's how we carry it around in our blood. Okay, so anemia, this is when there's low oxygen from lack of red blood cells, either low red blood cells being produced or the red blood cells are damaged and not able to transport oxygen as efficiently. So I'll let you pause here and look through this. In addition to misformed red blood cells or lack of red blood cells, we could have red blood cells that are being destroyed. Again, red blood cells have a relatively short lifespan. They live about 120 days normally. They're very small, and their job is to squeeze through the capillaries of active tissues. And squeezing through these very small capillaries will damage the cells. So even a healthy red blood cell will only live about 3 or 4 months. So we're constantly making more red blood cells. This is why being a blood donor is so important, because when people need blood transfusions, it takes a while to make more blood if you've lost blood because of an accident or something that you need. It takes a while to make more blood. So here are the steps of the destruction of red blood cells. And then this is this heme molecule. So this is heme right here. It's called a polyprotic 9 ring. You don't need to know that, but here in this ring is this iron at the middle. So Fe, this stands for iron. And inside here where the iron is, this is where the oxygen binds. So hemoglobin is made up of 4 regions. So this is a protein. It's called quaternary structure. It's got 4 very similar subunits, 2 alpha and beta subunits, 1, 2, 3, 4. And each of these little red looking things here, this is representing the heme. So this is hemoglobin, and each hemoglobin has 4 hemes, and heme can each pick up 1 oxygen. So you can transport 4 oxygens per 1 heme. Now later, when we talk about the urinary system and the digestive system, when we see how heme is broken apart, we're going to see the names of these molecules. Again, this biliverdin and bilirubin. This is how the heme is broken apart. This biliverdin is this structure with the iron, and this bilirubin is without. We're going to see this again. Biliverdin, verd, here this means green, and rubin, here this means like orangey yellow. This bilirubin is actually one of the things that gets excreted in our urine, and this is the cause of the color of our urine. So this is showing the steps of red blood cell formation. So the nutrients that we need are absorbed in our digestive system, and then that blood transports the absorbed nutrients to the red bone marrow, and this is the site of the hematopoiesis, or the making of the new red blood cells. From here, the red blood cells are made, and they circulate in the bloodstream for about 120 days. When the lifespan of the red blood cells is over, then it will be digested. It will be eaten by these macrophages of our own immune system, and it will break down the red blood cell into different pieces. It'll break down the hemoglobin into this globin in the heme, The heme will get broken apart into the iron and the biliverdin. The iron can then get recycled to go back to make more new blood. The biliverdin is converted to bilirubin, and then the bilirubin is what gets excreted through our urinary system. So this is the cycle that new blood cell formation goes through and how the old blood cells are broken apart. The iron can be recycled, and then the rest of that hemoglobin gets excreted. So that's red blood cells. So now let's move on to white blood cells. So white blood cells are much lower represented in our blood. We're going to see a table at the end that will show a breakdown, but there's many more red blood cells in a blood cell count than there are white blood cells. So white blood cells are also called leukocytes. The major job of white blood cells is in the immune system to protect against disease. The white blood cell hormones are interleukins or colony stimulating factors, which stimulate development of the white blood cells. There's 5 types of white blood cells that I'm going to go through quickly, and then we're going to compare and contrast them all at the end. So there's 2 major categories of these 5 types. There's called granulocytes and agranulocytes. And the granulocytes have granules that you can see in staining, and the agranulocytes do not. So that's how you can differentiate between these 2 categories. So this is the first of the granulocytes. This is neutrophils, and they have light purple granules when you do an acid-base stain. These are phagocytic, meaning they can eat other cells. And this makes up a good majority of the leukocytes. These are elevated in bacterial infections. And feel free to pause at any of these slides to get this information. Again, it's going to be all summarized in a chart at the end. Next are the eosinophils. These have red granules in an acid stain. This is sometimes seen in allergic reactions. It's 1 to 3% of the leukocytes. Also seems to be elevated in parasitic worm infestations. So for example, if you were sick and you went to the doctor and they did a white blood cell count, they would be testing how many white blood cells do you have total, and also what percentage of different white blood cells do you have. And if you had an elevated amount of eosinophils, that would be a clue to the doctor that either you're dealing with some sort of parasitic worm or an allergic reaction. So knowing the number of the different classes of white blood cells can be important in determining what sort of infection you might be fighting. A basophil, this is still a granulocyte. It has blue granules. This releases histamine. Histamine is released in inflammation responses. Inflammation responses causes our blood vessels to dilate, which means more blood is getting to the area of infection or whatever. It also releases heparin, which helps prevent clots and clotting. We're going to see more about that in a minute. And this is less than 1% of leukocytes. So those are the granulocytes. So now let's talk about the 2 classes of the agranulocytes. The first is called the monocytes. These are the largest of all blood cells. It is about 3 to 9% of our white blood cells. They can phagocytize bacteria, dead cells, and other debris. And then the last is called the lymphocytes. So these are slightly larger than a red blood cell, and these are actually the precursor to both T and B cells, which are both important in the immune system. We'll talk about these a lot more later when we talk about immunity. And the B cells produce antibodies, the T cells have other jobs, and this makes up 25 to 33% of leukocytes. So the function of white blood cells is to protect against infection, and we just saw how the different classes of white blood cells or leukocytes will focus on different types of specific infection. The way that they do this is that they're able to squeeze between the cells of the capillary wall and enter the tissue space outside of the blood vessel. This is called diapedesis, and this is how the white blood cell gets from the bloodstream to the tissue or the cell that it is trying to protect. White blood cells have enzymes that help them get into the cell or tissue that they are trying to get into. So let's look at a specific example. So here what we have is a splinter that's puncturing our skin, our epidermis and our dermis, and then it's come in contact with a blood vessel. So on this splinter, we have bacteria that are being introduced to the dermis. And when this happens, this stimulates a cascade of events for our leukocytes to come and neutralize this threat, kill this bacteria. So what happens is the bacteria that was on this splinter will multiply, and the injured cells around this splinter are going to release histamine into the blood. So this histamine is an injury response, and this causes the blood vessels to dilate, and when the blood vessels dilate, this is bringing more blood to that area. So with the blood, and the red blood cells also are the white blood cells, and neutrophils will move through the blood vessel walls and migrate towards the bacteria. And the neutrophils will destroy the bacteria by phagocytosis. And phagocytosis literally means cell eating. The way that it does this is these white blood cells have lysosomes in them, which is a cellular structure that's full of what are called hydrolytic enzymes. And these hydrolytic enzymes will kill the bacteria, dissolve their cell walls and stuff like that. So the functions of the white blood cells are to identify what the threat is, the bacteria in this case, and in this case, the neutrophils kill the bacteria. So a white blood cell count is a procedure that normally if you were sick, you went to the hospital, the first thing they might do is a blood panel where they would look at your white blood cell count and be able to tell if you are dealing with some sort of immune response. So it counts the number of white blood cells per cubic millimeter of blood, and typically it's between 5,000 and 10,000 white blood cells per millimeter of blood. So we'll see a chart that's going to compare all of this in a minute, but if you remember when we talked about red blood cells a few minutes ago, red blood cells were 4.6 to 6.2 million cells per cubic millimeter. So this is much, much less concentrated than the red blood cells. A low white blood cell count indicates that you could be dealing with one of these symptoms. Leukopenia is what that is called. You also could have leukocytosis if you have a high white blood cell count, which shows that there's an acute infection that you're fighting. So an infection could result in either a high white blood cell count or a low white blood cell count, depending on what type of infection it is and how your body is responding to it. Also, you can have a differential white blood cell count, which lists the percentages of the different leukocytes. So again, different leukocytes are responsible for fighting different types of infections, so that can give some insight into what type of infection your body might be fighting. And then this is a chart showing if elevated these different types of leukocytes, what that could be caused from. So you could pause and take a look at this. So the last type of cell that is in our formed cells of our blood are our platelets. Platelets are known as thrombocytes, and they lack a nucleus. They're about half the size of a red blood cell. There's a lot of them. There's about 150,000 to 450 per cubic millimeter of blood, so more than the white blood cells, but less than the red blood cells. And they help repair damaged blood vessels by sticking to broken surfaces. And thrombocytes or platelets also release serotonin, and the serotonin makes the smooth muscle walls of your blood vessels contract. So it contracts the blood vessel and also is helping to repair the damaged blood vessel. This is showing the different cellular components of blood. So red blood cells, white blood cells, and platelets, and the numbers and the functions. So this is a really good table for you to reference. It kind of summarizes everything that we just said. So you could push pause here if you wanted to look at this some more. Okay, so we said that the blood was broken down into two major components, the red blood cells, the white blood cells, and the platelets, and then the blood plasma. So the blood plasma is the liquid portion of the blood, and it makes up most of the blood volume, and it is 92% water. So if you remember back to chemistry, water is polar. We just talked about this a little bit with the hemoglobin need to transport oxygen, because water is polar, which means this is an aqueous environment where polar things are going to be soluble and dissolve, and non-polar things will be insoluble or not dissolve. So this is why we need these molecules, these proteins, these other cells to transport things that would not be soluble in our blood. So blood plasma is important for transporting nutrients, gases, vitamins, helps regulate fluid and electrolyte balance, and pH. So again, remember, homeostasis is the theme for all of these systems. All living things have to maintain these very specific parameters, and all of that is regulated to maintain homeostasis. So these are the most abundant dissolved substances. We call these solutes. Remember, in a solution, we have a solvent, and in this case, the solvent is water, and the solute is what's dissolved in the water. So these are the most abundantly found solutes in the plasma. So we have albumin. This helps maintain osmotic pressure. We'll talk about this more later. We have globulin. There's 3 types of globulin. The one that we'll probably talk about most are the gamma globulins, and the gamma globulins are the globulins that constitute the antibodies of the immune system. So IgG, IgA, IgD, IgE, IgM. I think those are the 5. We'll talk about that more later. And then fibrinogen. And fibrinogen is a precursor to protein fibrin, which is going to be responsible for coagulating blood and clotting, that we'll see a little bit later. So with gases and nutrients, we said there's 3 gases in the plasma: oxygen, carbon dioxide, and nitrogen. Again, nitrogen is the most prevalent, but it doesn't really play a physiological role, so we're not going to talk about it here. The other plasmas and the other nutrients in plasma are amino acids, which will be used to make proteins or come from proteins, simple sugars, nucleotides, lipids. All kinds of nutrients are transported in plasma. And then there are some non-protein nitrogenous substances. We'll talk about this more later. A lot of these are involved in waste, or some of these are involved in energy production and producing ATP and some of these high phosphate bonds. So we're going to come back to this when we see this specifically, and we'll remember to mention that these are transported in the blood plasma. Plasma electrolytes, these contain ions. So electrolytes are really just ions. So some of the common ones are sodium. Sodium and chloride are the most abundant. Potassium, calcium, magnesium. Bicarbonate is going to be really important later. This is HCO3-. Bicarbonate is important because it is necessary to help us get rid of the carbon dioxide. So we'll talk about that more later. Phosphate and sulfate. So we're going to see all of these electrolytes later, these ions. Okay, let's talk about how we stop bleeding if there is either internal or external bleeding. So the word that we use to describe stoppage of bleeding is hemostasis. Stasis means stop or static. Hemo refers to blood. And there's 3 major things that limit or prevent blood loss, and that's blood vessel spasm, so kind of contracting of the blood vessel, platelet plug formation, so we'll talk about platelets, and then blood coagulation. So the blood vessel spasm is triggered by pain receptors, platelet release, or serotonin, and the smooth muscle in the blood vessel contracts. So this is showing the platelet plug formation. This is triggered by exposure of platelets to collagen. Platelets adhere to a rough surface to form a plug. This is called cohesion. When these platelets start sticking to each other, there's cohesion and adhesion. Cohesion is what's happening here between these platelets. It's kind of like making a dam to stop the loss of blood from this damaged blood vessel. And then there's blood coagulation. There's two types of blood coagulation that we're going to look at in more detail, extrinsic and intrinsic. This is triggered by cellular damage. This is called extrinsic, is the one that's triggered by cellular damage, and blood contact with a foreign surface. This is called intrinsic. So cellular damage is extrinsic, blood contact with a foreign surface is intrinsic. In both cases, we're going to result in a blood clot, but the way that this cascade works is different depending on what started that response. So we're going to see a picture of that in a minute, but we will form a clot in both cases. This is just listing the three hemostatic mechanisms to stop blood loss, and you can pause here if you want to write this down. Okay, blood clotting mechanisms. So again, there's two types, and I'm going to show you this picture in a minute that's going to show us the two pathways. There's extrinsic, which is for tissue damage, and there's intrinsic, which is when blood contacts a foreign surface. In both cases, you are going to result in a fibrin clot. So what we're looking at here is a fibrin clot. So this protein here, fibrin, is sticking all of these blood cells together, and that is going to produce a clot and stop the bleeding. So again, there's two types. There's intrinsic and extrinsic. So extrinsic, this is in the tissue, and then extrinsic is in the blood. So here's intrinsic, and this is in the blood, and that triggers coagulation. Okay, blood coagulation, and then so extrinsic and intrinsic, and then these are the clotting factors. So I'm going to show you the cascade of events that are going to use these different clotting factors, and then you can come back to this if you want. So feel free to come back to this slide after we've talked about the next slide. Okay, so this slide is showing the difference between extrinsic and intrinsic. So extrinsic clotting mechanisms is tissue damage. So the factor 3 comes from the tissue. So it's not coming from the blood, it's coming extrinsically from the tissue that was damaged. This is called factor 3. And I'm not going to ask you specifically about each of these factors, but basically it's called a cascade. Most of the time in cellular processes like this, nothing is really one step. There's multiple steps, and we call these pathways or cascades. Ultimately, from this tissue damage, we end up with a fibrin clot. So if it's extrinsic, this means that the tissue was damaged and the tissue released factor 3, and the factor 3 started a cascade that resulted in the clot. If it's an intrinsic clotting method, intrinsic means that the blood contacted a foreign surface and the blood activated this Hagman factor, factor 7, and this now activates this cascade of steps that results in a fibrin clot. So in both cases, we're making a fibrin clot, but if it's extrinsic, it's the tissue that releases the factor 3, and if it's intrinsic, then it's the blood that contains this Hagman factor 7, and it activates this pathway. So in both cases, you end up with a clot, but depending on what originated this cascade, it just takes two different pathways. That's the big picture that I'm hoping you get from this. So once you've made this fibrin clot, there are a few different things that can happen. So the blood can retract and pull away from the edge of the blood vessel, and it can be digested, or it can form either a thrombus or an embolus. So a thrombus is an abnormal blood clot. An embolus is a blood clot that's broken off and now it's moving through the blood vessels. So you've heard of pulmonary embolisms or thrombus. These can be very dangerous. They can cause heart attacks or stroke when they are moving through blood vessels they're not meant to be moving through. Okay, the next thing to talk about is atherosclerosis, and we'll talk about this more later, but just while we're here in the blood vessels, you'll see here is a healthy blood vessel where you have the artery wall and the lumen, and the smooth muscle regulates the lumen diameter. This is where the blood would be flowing through in here, and if you have plaque that's built up, you see how it restricts and constricts the diameter that the blood has to move through. So this is atherosclerosis. Let's talk about some prevention of coagulation. So we can have sometimes when basophils and mast cells secrete heparin. This is an anticoagulant. Also, sometimes people who are at a high risk of forming blood clots can be put on medicine like heparin. And this will block some of the steps in that pathway that we looked at for the intrinsic and extrinsic clot formation. And that is being shown here. So if you wanted to take a look at this, you could see how that works. Okay, so the last thing that we have to talk about with blood are blood groups and transfusions. So there are a lot of genes, 31 genes, that are known to contribute to what are called blood groups or blood types. Basically, it's the type of antigens and antibodies that can be found on your red blood cells. So some terms that we need to be familiar with are agglutination, and agglutination means clumping of red blood cells together in response to a reaction between an antibody and an antigen. So this is bad. This would be like if you received a blood transfusion from a donor that had a different blood type than you that caused a reaction. It would form the new blood cells and your blood cells to clump together, which would be bad. So two other words we need to know are antigens. Antigens are they're proteins on the surface of the red blood cells that stimulate the production of antibodies. And then antibodies are proteins that react to specific antigens. So antibodies are usually found on immune cells, and antigens are found on the other cells, and they recognize each other in a very specific way. And we'll talk a lot about antibodies and antigens when we talk about the immune system. So let's look at the different blood groups. So there's A and B, and then there's AB, and then there's O. So there's two different antigens, two major antigen types on red blood cell membranes. There's antigen A and antigen B. So if you are a blood type A, that means you have the antigen A and you have anti-B antibodies. If you are a blood type B, that means you have the antigen B and you have anti-A antibodies. If your blood type is AB, that means you have both A and B antigens and neither anti-A nor anti-B antibodies. And if you're an O blood type, then you have neither A or B antigens and both A and B antibodies. So the way that looks is here, this is what A looks like. It has A antigens and anti-B antibodies. Type B has B antigens and anti-A antibodies. AB has A and B antigens, and then type O has no antigens and both A and B antibodies. So what that means is if you put two blood types together that are not compatible, it will form this agglutinate, which means the blood will clump together. So if you are type O, then this means you're a universal donor, so you can give blood to any of the types. If you are type AB, that means you're a universal recipient. You can receive blood from any type. So you also might hear something about being AB positive, A positive, something like that. So where does that come from? That comes from this Rh blood group. And this Rh is named for the rhesus monkey, where it was first studied and learned about. And this is testing for the Rh antigen. So this is just another antigen. If you are Rh positive, that means you have the antigen. This is antigen D or other Rh antigens on the cell membrane. So if you are Rh positive, then that means your whatever other antigens you have, A, B, O, you would be that and a positive. If you don't have those antigens, then you would be Rh negative. So the anti-Rh negative antibodies form only in the Rh negative individuals and in response to the presence of anti-Rh cells. So the consequences for this is it can cause what's called erythroblastosis fetalis. This is a blood disease in newborns. This is something that you would be tested for if you were pregnant. The problem is if you have this isn't a problem in a first pregnancy, but if you were to have a baby that had a different Rh type, so if you have an Rh negative woman and an Rh positive fetus, the cells from the Rh positive fetus can enter the woman's bloodstream, and the woman becomes sensitized to the antibodies and can fight the Rh positive cells. And then in the next pregnancy, if she had an Rh positive fetus, her own Rh positive antibodies would fight the antibodies in the fetus. So this is something that can be treated when detected, not a problem for first pregnancies. This is something that they normally test for with subsequent pregnancies to make sure that there is not a conflict between the antibodies in the mother's blood and the fetus's blood. That's it for blood. I'll see you next time.